Today, we're going to talk about statistical analysis plan, SAP. I think for most of the clinical statisticians, uh, you should be aware of this document, SAP. Uh, but before I get into what SAP is about, I want to give you a little historical perspective. Um, I joined the industry in 1987. At that time, there's no such a thing as SAP. And so if you look at the protocol in the 70s and 80s about the statistical methodology, uh, in the protocol, it just says appropriate statistical method will be applied to analyze the clinical data. So there's no pre-specified way of alpha protection, and there was everybody was kind of wait to see the data. The clinical trial is over. When the trial is completed, then we jump on to perform all kind of analysis. So I remember in those years, before any clinical trial is unblinded, there's not much we can do. I mean, we're basically just waiting for the last patient out and waiting for the database to be cleaned, to be locked, and then unblind. Once unblind, everybody jump up and perform analysis. uh, so we don't really have such a thing called primary analysis or secondary analysis or anything like that. We just analyze the data from left to right, and the entire clinical team is very eager to know the results. So I remember that I was sitting there and performing analysis, and the team members come in to my office, says, "Night, how did it go?" So while I was running the program, I sort of print out some of the just rough draft kind of results. And then they looked at it, they're very excited. They say, well, 90, this looks good. Let's do this, let's do that. So before finishing my first job, I was jumping into the second job, the third job, and just repeat a lot of different kind of analysis, different requests. And I sort of overlooked some of the details. So in my SAS code, I probably did a wrong code or wrong logic or something like that. So by performing these analyses repeatedly, you check with the previous results, and I realized I made a mistake. So I have to go back to the requester and said, sorry, the tables that I gave you earlier was not right. I made adjustment now. And so it was just the thing you worry about is last patient out and database unblind. Once the data are unblinded, I began to run and very busy and trying to answer a lot of requests and making mistakes and all that kind of stuff. So that was pretty much the late 80s, early 90s. That was my career as a clinical statistician. Is just whenever trial is unblinded, you be very busy. And before the trial is unblinded, you're working on other stuff. In early 90s, I believe it was 92, 91, 92 sometime, the management was circulating a memo. So I take a look at memo. The memo says there's something they call SAP. They describe what SAP is. As soon as I see that memo, I said, this is the way to go. Yeah, we should do a statistical analysis plan, and with such a useful tool. I believe the whole industry bought into this idea of SAP, probably just within a few months. It's not just within Pfizer, where I was at Pfizer at that time. Every other companies kind of looked at the idea of SAP. They say it makes sense. Now, let me explain why it makes so much sense to me. All right. For many of you who will be working in industry, or for those who have been in the industry for some time, you are aware of that in industry, the key survival rule is to manage the expectation. All right. So in other words, you don't want people to have too low an expectation of you. You don't want them to have too high an expectation of you. I mean, the earlier part of my career, I sort of set the expectation too high because everybody says 90 help. I say yes. 90 help. I said yes. And people just thought I can do everything, but I couldn't. Now, the biggest advantage of SAP is to manage the expectation. Because in that SAP, you sort of specify what kind of tables you're going to produce. And then the team will not expect you to produce more. And once you have this SAP on hand, you can negotiate, you can bargain. If they want more tables, you say, I will need more time. But without SAP, you don't have any basis to bargain. So the biggest advantage of SAP, to me at that point in time, was it serves as a contract. This contract will help us negotiate timeline, negotiate deliverables, and will help us with resource allocation and all those kind of stuff. This is a wonderful document. Let's do it. You know, SAP just makes perfect sense. So we started working at SAP. Only after a few years that I had more experience with SAP and things like that. And then we realized, well, we can use SAP to demonstrate to FDA that our alpha is protected. So this is to me at that point in time was a totally new realization, a new understanding. Oh, there's something about alpha protection, and we can use SAP to demonstrate the alpha is protected. But in order to do that, there has to be a documentation system. In other words, you're not only going to show FDA to say, here, look at this document, we've signed off SAP on day such and such, which is before breaking blind. But you have to have a very strict, a sort of in the highly regulated way within the company that there's a documentation system, that this system would prove that the signed off SAP has been checked in on that date, and that date was before breaking blind. So during the 90s, from the statistical group, the clinical statisticians, we started working on SAP. And then we started working with the regulatory affairs group, and then we come up with this documentation system, and of course the software and everything like that. So now, if we have an SAP and we have a documentation system, then we can demonstrate to any regulatory agency, not only FDA, but also EMA and any foreign countries, to show them, to say, we've already agreed on the primary analysis method and the model to be used and everything like that before breaking blind, so that the alpha is protected for the study. So with these two understandings, I hope you can understand that the two objectives of SAP is, first, it serves as a contract. It's an internal within the company, within the team across different functions, as an agreement of what needs to be delivered. And to external, to any of the regular agencies, we were able to use SAP to demonstrate the alpha is fully protected. So that is the purpose of SAP. Okay, let's get started. So we're going to learn what should be documented in protocol, statistical analysis plan, and clinical study report, CSR. All right. If you are a clinical statistician, you know that the three tangible deliverables as a clinical statistician are these three documents: protocols, SAPs, and CSRs. want to understand the statistician's role in developing protocols, SAPs, and CSRs, discuss pitfalls in developing these documents. I want to come back to this point about tangible deliverables. To me personally, I think the most important contribution a clinical statistician make to the entire organization are two things, all right? Study design and statistical consulting. Both are kind of intangible, or you can say study design can be reflected on protocol, so it was somewhat tangible. People can see, NITU designed this protocol. But that protocol design reflects not only a study design, but a lot of communication, mutual understanding, compromise, and deal with practical issues and all that kind of stuff. So study design and statistical consulting are the two most important contributions a clinical statistician can make to the organization. But if you want to make it tangible, then these are the three documents: SAP, protocol, SAP, and CSR. Because by the end of the year, when there's time for performance appraisal, I can go to my boss and say, look, this year I did so many SAPs, so many protocols, so many CSRs, and they can count it. I mean, it's tangible. But the most important contributions are intangible. All right, three documents are created that contain description of statistical methodology: protocol, SAP, and CSR. So let's start with protocol. All right. Now, in a protocol, there is a statistical method section. Within the statistical method section, there are two most important things. One being sample size justification. All right. As I mentioned previously, that sample size actually drives the entire study design. The entire thinking about sample size will guide how you design the protocol. Because sample size, so what are sample size? Sample size is a function of alpha, beta, delta, sigma. Right? Alpha and beta reflects the risks the team is willing to take. All right, alpha is risk of developing a placebo. Beta is risk of giving up a good drug. So when there is a signed up protocol, in that protocol there's a sample size. That sample size reflects the risk the team is willing to take. And delta and sigma reflects the assumptions the team is willing to make. You know, how big a delta, how much treatment difference, what kind of standard deviation you're dealing with. These are some assumptions we're making. So sample size is really the fundamental thinking of the entire trial design. All right. So protocol, you have to have sample size justification. You have to describe fully, clearly as to why you choose this sample size. What kind of alpha, beta, delta, sigma that you're choosing, and the assumptions you're willing to make, and the risk you're willing to take. All right, so protocol includes a detailed and comprehensive statement of the objectives and the trial and the statistical method to be used in order to meet these objectives. All right, objectives is absolutely critical, absolutely important. All right, every trial always starts with a clinical question. So what is the key question? How do we translate this question into an objective? And how do we translate this object into a hypothesis? Okay. So again, like I said, in the protocol, there is a statistical method section. In the statistical method section, there are two components. One component being sample size justification, the other component being the primary analysis method. The primary analysis method really helps you to demonstrate how alpha is protected. Right? This alpha protection is reflected from your primary endpoint, your primary comparison, your primary analysis set, primary time point, and whenever there's any multiple issues, multiplicity issues, you need to make alpha adjustment. So all of these critical thinking will have to be reflected in the protocol, in the statistical method section. All right. So in the statistical method section, first thing is sample size justification. Why do we choose the sample size? Of course, this is corresponding to your study objective, and the objective is translated into statistical hypothesis. And the statistical hypothesis will guide you how to analyze the data, and then how can you ensure to all the regular agencies that the alpha is fully protected. SAP. SAP is a comprehensive and detailed description of methods and presentation of proposed data analysis in order to avoid post hoc decisions. It's more detailed than protocol. Actually, the protocol gives you a guidance. Protocol gave you, in the protocol, the statistical method section says, here's the objective, here's the hypothesis, and here's the primary analysis method, and all that kind of stuff. So that it has to be up to a level that everybody sees that statistical method section in the protocol, they can be convinced the alpha is protected. But given that, it is not detailed enough to reflect what the real data could possibly be. Let me give you an example. You can say in the protocol as to a particular model you're going to use. And then after the study going and the data came in, and of course we do blinded data review. When going through blinded data review, you realize the actual data is somewhat different from what you were thinking of at the time you're designing the protocol. So you may have to make some adjustment to your statistical analysis. And then whenever that happens, you would like the SAP to reflect the newest way of looking at your statistical method. All right. So SAP is not only more detailed, but also based on some practical experiences after you've seen the blinded data review. So you do want to allow that wiggle room. So you can get the protocol started because you have the sample size, you have the primary analytical method. Those are pre-specified in the protocol. You're done. So the protocol started ongoing. And then now you start to think about secondary analysis, supportive analysis, exporter analysis, sensitivity analysis, start thinking all of this. And these kind of things will go into SAP. They don't have to go into the protocol. Protocol doesn't have to say anything about how you're going to perform exporter data analysis. That's not necessary. Well, even for secondary analysis, it's not strictly necessary because in protocol, the key concern is about alpha protection. Now, still, SAP, the key concern is alpha protection, all right? All right, your SAP can be very fancy, but if the SAP does not protect alpha, it's a piece of junk, okay? So in SAP, you have to be very clear about how the alpha is protected. Later, I'm going to talk about the detail about the SAP. Basically, SAP, there's 3 documents. There's the text part, which is the SAP, what we write, all right? And there's a table of contents. The table of contents will be what are the tables you're going to produce for the study report, all right? And the third component being table templates or mock tables. CSR. CSR provides a detailed description of the trial results and the interpretation of the results. Reviews all planned statistical methods and describe any deviations from the plan. Writing CSR, writing a study report is a very, very important practice for any of the statisticians. We learn so much by writing this CSR. If you look at the clinical trial data, every trial, after unblinding, you start to perform analysis and from left to right, right? You do a lot of analysis. Why do you want to perform all these analyses? You want to learn what does the trial tell you, right? I mean, the company spends a lot of money to run the clinical trial, sometimes hundreds of millions of dollars to run just one trial. And they invest so much money into and resource into this trial. And like I said, all these investment and resources come back with data. The data must tell you some kind of story. No matter the drug is successful or the drug is failed or whatever, there is a story from the data. As a clinical statistician, it's our job to understand the story the data tells us. So we need to digest all these data. How can you digest these data? You perform analysis. You do your performance. You do your primary analysis. You do your secondary analysis. You look at primary endpoint, secondary endpoint. You look at even subgroup analysis, exploratory analysis, sensitivity analysis. For all of those, you try to understand the data because the data is telling a story. So it is our job as a clinical statistician to understand what the data is telling us, what the story is about. And the writing of the study report is about translating the story the data tells me into a story so that other people can understand. All right. Now, there are a lot of things we should be thinking about when we write a study report. And when I teach, I told my students, practice writing is so important because I think one of the most important part of our work as a clinical statistician is about communication. Many years ago, there was a summer intern came to work with us, and she came in my office and says, "Nike, I'm eager to find out what do you guys do in industry." I said, "We do 4 things. We read, write, listen, and speak." And then she thought about it. She said, "Well, we do that in school also." So I said, "Yes." So that lets you know how important communication is. It's really about communication. And I would add to say, in school, you'll probably do more reading and listening, less about writing and speaking. But once you come to the real world, especially you work with the industry, writing and speaking is absolutely crucial. It's very, very critical. Practice writing, practice writing, practice writing. So when I teach writing, before you start write, what should come to your mind first? All right. The first thing you should think of is, who is your audience? Who are you writing this report for? What do you want the report to achieve? We all know writing is a lot of effort. Like I said, you have to understand the story the data told you. You have to reflect the story. You have to write it. You have to express it, be able to demonstrate it. It's a lot of work. Now, if I do a lot of work, what do I want to achieve? And people say, yeah, we want the reader to understand. Yeah, I learned the story. I want to share the story. They should be able to understand the story. Is that sufficient? No. If we spend so much effort in writing the report, that's definitely not enough. I not only want my reader to understand, I also want my reader to take the action that I ask them to take. So what action? All right. So now I look at the study, I look at the data, the clinical trial. I'm blinded. I perform the analysis. I learned the story about this trial. The data told me this. The story should be very clear to me. Right, the first question I ask is that, should I continue to develop this drug, or should we submit this drug to FDA or whatever? That means, is the drug successful in this trial? If it is successful, now I know I'm writing this report to FDA, because if it's successful, eventually it will go to FDA or go to any regular agencies globally. All right. If my audience is regulatory agency, and I'm convinced the drug works, then I want people to read my report and be comfortable to approve this drug. But if I look at the story from the data, clearly the drug does not work. Then my report is for upper management. I need to convince the company, do not waste more resources on this drug. Okay, so the way you write is driven by the way you think. You have to understand who your audience is, who the readers are, and then you have to write your report to lead your readers into what you want them to do. Right? They're not only going to look at your report and they say, okay, Nike, I know what you're talking about. No, that's not enough. They also need to know what the report tells them, and so they're going to take the corresponding action ask them to take. So that's how you write. So let's get more details into protocol, all right? What contributions does the statistician make to the development of the protocol? All right. Understanding the objective. This is absolutely critical, important, all right? Specify trial objectives with clearly defined endpoints, all right? We have to understand the objective. We have to have a very clear understanding of the objective. Well, most people say, well, you look at the draft protocol where the conditions already put in there says the objective of the study is to compare the efficacy and safety of the test drug against the control. Well, that's a statement. Is that statement clear enough to you? Do we fully understand what does that mean? So first of all, what is the disease? What is the condition? Right. What does it mean by efficacy? What does it mean by safety? Okay. Now, how to measure? All right. So how can we translate that study objective into a statistical hypothesis? All right. And from that statistical hypothesis, it reflects the endpoints. And of course, nowadays we're talking about estimate. So what is your estimate? We need to understand what that is. And how do I achieve that estimate? All right. And then how can I write it as a statistical hypothesis that is testable? Okay. And then from that statistical hypothesis, of course, that corresponds to the endpoint. And before you calculate sample size, you need to know the delta and all the details associated with it. So you have to have a very clear understanding of the objective and translate into a statistical hypothesis. Right? Definition and measurements. All right, definition and measurements of endpoints. Methods of randomization to treatment groups, sample size justification. That's big. All right. Sample size is very, very important. All right. Specific parameters to be estimated and statistical hypothesis to be tested. Primary analysis population. So what is our primary analysis population? All right. It should be intention to treat, right? And of course, we define the full analysis set, which is a practically achievable way of implementing ITT. All right. General strategy for dealing with each efficacy variable. What we do generally in a protocol, usually they have, any of the clinical trials, if you look at the endpoint, there's 3 data types. There's continuous data, discrete data, and time to event data. So what we try to do is that for a given protocol, or even for a given drug, all the protocols associated with that drug, we would like to have a consistent way of analyzing continuous data. So if your primary analysis of the primary endpoint is analysis variance or analysis covariance, then you want all the continuous data will follow the same kind of model. All right, you maintain some kind of consistency. Now, if it's discrete data, and my preference of analyzing discrete data will be Kirkland-Hanzel. So I will probably apply Kirkland-Hanzel for all kinds of discrete data within the same protocol and within probably multiple protocols within the same drug. Okay, so maintain some kind of consistency. Primary analysis, population, general strategy, plans for interim analysis, if any. I mean, I think that's a big deal. I mean, if you have interim analysis, and of course we do adaptive design and the other things. All right, we be very careful about those. Plans for sample size revision if needed, and review of CRF. CRF is important. All right. Usually statisticians do not pay too much attention to CRF, but CRF is critical. That's where we get our data. That's the source of our data. All right. The two things to keep in mind in any of the CRF design process. All right. The two things is do not collect too much and do not collect too little. All right. So everything you collect, you have to know clearly what that piece of data is going to be used for. All right. If it's not needed, it's our job as a statistician to convince the team not to collect it. All right. Now, if you work with a clinical team, the team just have a tendency to over collect. All right. At the designing stage, it's our job to warn them, do not over collect. All right. If because for every piece of data you collect, you have to design the CRF to collect it. You have to physically collect that piece of data. And that piece of data will be stored in the database. And we have to issue queries for that piece of data. And we have to clean up that data. And eventually, we need to extract that data from the database, and we need to report that data. We need to list it. We need to analyze that data. For every piece of data we collect, there's a lot of resource involved. So if that piece of data is not relevant to the objective of the trial, do not collect it. Okay. Of course, there's key data you have to collect, so don't miss it. Usually, that won't be the case. In most cases, people would have thought about and have all the necessary data to be collected, but people have a tendency of over-collect. So it's our job to make sure it's not over-collecting. Things to do before writing the protocol: talk to the study team and key partners, understanding the medical condition, understand the measurement scales, understand the trial, perform a literature review, know the FDA expectations, know the company's SOPs. SOP being standard operating procedures. All right, study design is one of the most important contributions a statistician make to the organization. All right. Study design is not only critical from data collection, data analysis, and all that kind of point of view. Remember, previously we talked about CDP, clinical development plan. In the CDP, every protocol should have its position. All right. So when the team came to me and says, "90, we need to design the protocol," my first question is why? Why do we need to design this protocol? I want to see the CDP. Where does this protocol fit? Is it phase 1, phase 2, phase 3? And before designing this trial, what information do we have? How much do we know about this trial? And then after the trial is completed, what questions we need to answer? All right. So we need to have a clear understanding of where it came from and where we want to arrive at. All right. We have to have a clear understanding. Why do we need to do this trial? And what do we want to achieve from this trial? And of course, then we need to understand the medical condition, understand the measurement scales, understand the trial and everything like that. To me, the entire drug development process, the entire protocol design and executing and analysis stage is a constantly learning process. I'm always learning. Either it's a new disease condition or different form of drugs or a different endpoints. You're constantly learning about this, and this learning is a two-way learning. It's not only I'm learning from all these experts and these different specialties, but also I need to communicate with them. I need to let them know what statisticians are thinking. What are I mean, there's statistical ethics. What are statistical ethics I need to follow and all these kind of stuff. So it's communication, learning, and it's mutual learning, mutual education. Reviewing CRF and other data sources. Poor data collection cannot be rescued by fancy analysis. Don't collect too little or too much. All right, whatever you collect, collect correctly. Do understand every piece of data you're collecting. All right. Data often collected by staff with low statistical expertise. Many pitfalls are associated with not finding out exactly how the data were collected and applying statistical procedures based on incorrect assumptions. All right, know your data. That's absolutely critical, absolutely important. Of course, the CRF should reflect drug efficacy and drug safety. All right, does the drug work? So, as we said, there's no way to objectively say a drug is efficacious, other than the statistical hypothesis approach to say the probability the drug does not work is controlled under alpha, right? So hypothesis test, does the drug work? What's your null hypothesis? What's your alternative hypothesis? And of course, alpha control. The entire trial is about alpha control, right? Regulatory agencies focus on control of type 1 error. Probability of making type 1 error is not greater than alpha, so alpha protection. All right. For phase 3, half alpha being 0.025 or 1 in 40. All right. And we're to avoid inflation of this error. So this is how you pre-specify in your protocol as to what you do to make sure your alpha is protected. And we know change of method of analysis to fit data will inflate alpha. In other words, the story I told you when I first entered this industry, we're talking about appropriate statistical methods will be used to analyze the clinical data, and there's no alpha protection at all. Right? So you cannot sort of look at the data and try to find a model to fit it. Don't do that. You have to 100% completely pre-specify how you're going to analyze the data. And of course, model comparisons, we talk about multiple endpoint, multiple treatment groups and things like that. So for 40 independent endpoints, one being significant random. I mean, if you designing a protocol with 40 efficacy endpoints without any alpha adjustment, and then just by chance, one being significant, it's just purely chance situation, right? 1 in 40. 40 independent treatment comparisons, small studies, multiple comparison adjustment. We can get into a little more details about multiple comparison, but I don't think it's really necessary. All right. So other types of multiple comparison, compare test drug with placebo and active control, multiple endpoints, subset analysis. There are different statistical methods available to handle these situations. So we know there's a lot of statistical publications dealing with all these kind of scenario. So if there's an interim analysis, we need to plan for it, right? Now, what is the final analysis? After the last patient visit, you close the database, you break blind, and you perform final analysis. So any analysis before last patient visit is interim analysis. So what's the objective? All right, you want to claim efficacy, you want to stop for efficacy, help decision making for other studies, stop the drug for no efficacy, all right, or other objectives. So control of type 1 error is actually critical because that's the foundation for drug approval. So there's experiment-wide type 1 error is controlled by specifying primary endpoint, primary comparison, primary time point, primary study population, and keep analysis methods as stated in the protocol. If interim analysis is needed, we should plan for it, pre-specify for it. SAP, okay. So like I said, SAP is a contract. within the company, within the team, is a contract of what you're going to deliver. So we use this document to help manage the expectation. All right. SAP is more detailed and technical than protocol. Should be consistent with the protocol in terms of the primary statistical method. The main thinking should be the same, but the actual analysis method could be vary. You can differ a little bit from what the protocol said, because by the end of the day, it's SAP, whatever specified in the SAP, that matters. Okay. May include templates for analysis display. So like I said, SAP, there are three components. There's the text part, which is the SAP itself. And there's the table of contents for the CSR. And also there's the table templates. Approved by study team and sign off before the blind is broken. Once approved, deviations should be described and fully documented. So here's how I write an SAP, all right? Writing an SAP doesn't take really much time for me to do, all right? But what's the most time consuming is this discussion, all right? So before I draft my SAP, I want to talk to the team. I want to talk to the regular affairs. I want to talk to clinical. I want to talk to the programming group. I will say, here's how I plan to analyze this trial, all right? And these are the things I have in mind. And can you agree with this, what we're gonna do? And when I talk to the program and data managers, I wanna make sure that for the data we collect from CRF, and I need to do this calculation. Your data, the way the data structure, the way you put the data sets together, you can support this kind of tables. All right, so I need to talk to all these people to understand what their requirements are and what are the constraints, what are the limitations and all that. And then it's a compromise, okay? In a way, you can look at the clinicians, the medical writers, and the regular affair people. These colleagues are the table users. So when you produce these tables, they're going to use these tables for writing reports, to communicate with upper management, legal agencies, kind of stuff. And the programmers and data managers, they are the table producers. Of course, the main people would be programmers. I truly, truly value the programmer input. And they're a very important resource for me because they're going to deliver the table. So I'm serving as a middleman to prepare this contract so that the table users fully understand what they expect. And then the table producers fully understand what they can do, what they need to do, all right? So this is what the SAP is about. So only after all these communications, then I came back and sit down and draft my SAP. So after I draft my SAP, I sort of circulate. All the key players get to see it. And then as a SAP meeting, and everybody agree, and I'll incorporate all the comments, and then I finalize the SAP, and I check into the documentation system. All right, once you check in the documentation system, now it's a done deal. All right, that means the SAP is signed off, it's already done there. Of course, there are going to be revisions, all right? And there are going to be changes, even though after I lock down the SAP, I already checked in the documentation system. There could be change of ideas, there could be practice situations, and you need to make updates to the SAP. So many statisticians would have to face the dilemma, whether should I check out this SAP, make changes to SAP, and recheck in, or should I document the changes from somewhere else? All right, so here's my advice. All right, think about the change, whether the change is relating to alpha or not. If it's relating to alpha, sorry, you have to check out the document, make changes, and rechecking again. It's a lot of extra effort. All right, but if the changes has nothing to do with alpha, you can put in meeting minutes, and in the later stage when you write this statistical report, you can include these changes into the statistical report to describe what happened. Okay. Give an overview of trial and restate the objective, clearly describe the statistical hypothesis and methods of testing that will ultimately determine whether the study is success or failure. And consideration should be given to each of the following levels of clinical and statistical significance, methods of dealing with multiple endpoint, missing data, outliers, withdrawals, use of baseline values and covariates, subgroup analysis, methods for handling multi-center data, interim analysis, stopping rules, concomitant medication, rules for calculation of derived variables, issues surrounding multiplicity, methods for testing statistical assumptions. I don't do that nowadays because most of the time the statistical assumptions would fit okay. All right. Exposure analysis. Other things to keep in mind: only results of analysis in this stage from the protocol can be regarded as confirmatory. So hypothesis testing, I mean, alpha protection, that's key. All right. Provide enough detail so that a reasonable person could reproduce the analysis. It's not only that when I change hand, you know, there are many times in the company, since IT, we have another urgent project, you have to work on that. So I was in the middle of writing the SAP, I have to pass on my SAP to the next statistician. And this statistician can pick up what I said, understand what I'm trying to do, all right? Not only that, but also, we know that FDA, they have the capability of analyzing the data. So we need to be clear enough so that the FDA statistician look at what I specified, they can reproduce my analysis, okay? Allows transfer study to other people. If analysis is complicated, you should specify computer code in the SAP. Yeah, there are times I just put the SAS code into it so that the programmer would not be confused. I mean, the times could be somewhat complicated, so I just put my computer code in it. It's a contract for allocating resources and division of labor. All right, so I told you that. I mean, that's the first objective is serving as a contract. Be aware of possible sources of bias. Work closely with clinical trial monitors and data managers to make sure you understand how the data are collected and cleaned. The extent to which the procedures in the protocol are followed and the primary analysis plan a priori will contribute to the degree of confidence in the final results of the conclusion of the trial. So you want to make it very valid, very professional, so that people look at it, they can be convinced. Guidelines. So these are some kind of table templates, you know, patient characteristics, demographic data, and all that. Analysis displays tables, graphs, listings. Problem with analysis displays insufficient titles or footnote and all these. All right. Problem with analysis displays. Goal of statistical analysis is to present the data in such a way that most readers will believe the conclusions drawn. It is very important to put information regarding statistical methods in the SAP and have the final version signed off before blind is broken. Yeah. SAP has to be signed off way before blind broken. CSR. All right. I want to just give you an overview is about a CSR, a clinical study report. All right. If you go to the medical writer or you go to the physician to say, what is a perfect report? All right. Now the physician will tell you, Nike, a perfect study report is a complete report. In other words, you leave no stone unturned. You want to be able to study completely and have a clear understanding, and that's your report. And I mean, that's the way they are trained to think. I mean, physicians, they went to medical school, they're trained to be complete because there's a patient in front of this physician, and the physician before making diagnosis, need to sort of understand the best of the patient, the patient current situation, patient's history, even patient's family history, and whatever tests we did and stuff like that. And so they have to have a complete understanding before they make the best diagnosis. That's how they were trained. All right. Now that training is very different from writing a CSR. A study report actually reflects the findings from a population, not from an individual. Okay. So what is a perfect study report? So here's a perfect study report. A perfect study report is like a statue. You cannot take out more. In other words, it has to be simple. It has to be clear. It has to be understandable. There's never a complete study report. There's never anything can be complete. But a perfect report is that if you take anything out, you're missing something. For my study report, it's absolutely lean and mean. There's no fat. All right. Everything need to be there, has to be there. Everything do not have to be there, it's not there. It should not be there. That's how you prepare a study report. All right. Now, another part of it is that in the past, before we have medical writers, I write the statistical report and the clinician writes the medical report, and we combine these two reports. And then, once you combine, it looks like two reports. It's kind of difficult. But now we have medical writers, so that's not a non-issue nowadays. So remember, CSR has to be lean and mean. So if you look at SCHE3, it gives you some guidance and structures. CSR is a truly integrated statistical and clinical report. Like I say, in the past, it was kind of two different people write it. Nowadays, not an issue. The report was appendices should provide enough individual patient data, including demographics, baseline data, methods that allow replication of the critical analysis for authorities to replicate your results. There's always this results review meeting. So this happens after I have my statistical analysis. In other words, the data is telling me some story. So when I walk into this results review meeting, I already know the story. And in that meeting, we'll discuss, the way I see the data. And of course, the team will reflect their way of seeing this data. And then how do we tell the story? How do we have a integrated way of understanding the story? And then we write this report, the report will have to reflect the story. So project team meeting held after statistical analysis has been prepared, statistical review primary results with study team. They jointly decide how to proceed. Write simple, clear English using short sentences. If you're having trouble getting started, jot down the points. So here's how you write. You look at the story, you look at the data tell you a story. The story should be very clear in your mind. You look at the data, any way you analyze it, it gives you a story. Now, it's our job to reflect the story from data to our readers. Okay. So think very clearly as to what is the storyline. All right, in a way that there's only one main story and you want to tell the story, right? But before leading the story, you need to give the background, right? And then after telling the story, you need to be able to wrap it up. So that's how you write the study report. And then start with bullet points, how you want to tell the story. So I said, do not assume your reader knows more than what they actually did. So you have to provide sufficient background. Give extra attention to presentations of graphs and tables. Review the report several times and get it reviewed. All right. Lots of pressure to do analysis quickly. People will remember the quality and often forget the speed. So we are the gatekeepers of the entire drug development process. All right. And it's our job to make sure that we understand what the data tells us, what the story is about. Thank you.